This is a production of Cornell University.
So today my talk is about grape genetics and grape genomics. So I had to admit the thing is I never actually work in the vineyard. So most of the work I did is on the computer. So the work I'm going to present today is mostly a collaboration with this guy shown here, Lance Cato Davidson. He's a USDA ARS scientist, based in Geneva, New York, and the computational analysis I'm going to show today mostly done by Chen Zhou and Dustin Wilkinson in my group. And the data I'm going to use, data I used mostly contributed by the breeders participate in the we call Vitagen project. Vitagen basically is the USDA NIFA funded project. The design of this project is we work with breeders in the US and we provide breeding service to the genotyping service mostly to the and the Lance also provide phenotyping service to the grape breeders in the US. And in return, breeders allow us to use their data to build computational models and build a computational method. So it's a win-win situation to help each other to work on the grapes. And so today's talk, I probably use, sometimes you will hear me say, we did this, we did that. And so that I just want to start with clarify who are the we when I'm talking about we. So first I would like to give you a brief overview of the genetic diversity in grapes. So grape is in the genus called Vitis. This Vitis, it's emerged about 20 million years ago. Started for a couple of years ago, the Vitagen project collaborate with Breeding Insight led by Mario Sheehan's group, Mario Sheehan, and the two national grape repository, USDA grape repositories, one based in Davis, California, and the other based in Geneva, New York. So we basically three groups team together. We start to genotype all the accessions from the USDA repositories. So far we have genotype more than 7,000 accessions from the repositories, and we just got our last batch of data last month. So the PCA plot I'm showing here has not included the latest batch of data, but should represent most of the genetic diversity in the USDA collection. So if you look at this PCA plot, if you look at PC1, which is the x-axis, so if you look at the x-axis, we can see like the grapes are separate in two genera, subgenera in Vitis genus. On the left side, this cluster of green dots are in the genus called Muscadina. So these Muscadina grapes are the native to the southeastern US and from North Carolina all the way to Florida to the south in the south and to Arkansas in the west. So basically mostly they adapt to the very humid, hot weathers in the southern US. So they are very different. They look very different from the grape we are used to as shown here, right? So they are very different. So one distinct, what we will notice immediately, their berry is a lot bigger. It's almost look like a plum, and they are very thick skins and very distinct, very strong musk flavor. So they are very different. They look very different from regular grapes we used to in the grocery store, and they have 20 chromosomes instead of 19 chromosomes in the regular grapes, or we called bunch grapes, or they're in the subgenus called the Uvitis. So what's on the right are the subgenus called Uvitis. So they look very different, but however, the breeders able to make cross between these two very different, even though they have different chromosome numbers, breeders still able to make cross. They're difficult, but still possible to make cross between the Uvitis and Muscadina and produce fertile progeny. So, and this has been served a great resource for plant breeding, for grape breeding, because this Muscadina, they adapt to the humid, hot weather which are very resistant, many different kind of fungus. So the genes, the fungus resistant genes from Muscardina has been served a great resource, genetic resource to improve the uveitis grapes to help improve the resistance trait. So if I zoom in just into the uveitis clade or uveitis subgenus, you will see that it forms distinct groups clustered by geographically. On the left side, there are a couple of species like Cinerea, Lupestris, Riparia. They're native to North America. On the middle is a species called Amurensis, which is native to Asia. On the right, you will see white grape is a species called Silvestris, and the Silvestris grape is the direct ancestor of the Vitis vinifera. So Vitis vinifera is a domesticated grapes. A distinction between a big distinction between the domestic grape and the wild grape is that wild grape relying for the wind pollination, and the domestic grape in Vitis vinifera is self-pollinated. It has perfect flower, has both male and female, and it's self-compatible. So one thing is in domestic grape is because this self-pollination, the efficiency of pollination much better than the wild grapes. So what you think you will notice in the wild grapes, the cluster not as dense in the domestic grapes. You can get really dense grape because this efficiency and fertilization. So that's one of the major steps during the domestication, grape domestication. So if I zoom into the Vitis vinifera clade in this PCA plot, then you start to see some of the names you're familiar with, like Riesling, like Pinot Noir, Merlot, and Cabernet Sauvignon, some wine, very famous wine grapes. So in this PCA plot, you can see the wine grapes is showing red on the right side. So, and the table grapes are showing blue on the right side. So actually you can see the PCA, the first PC can separate the wine grapes and table grapes, basically that breed for very different characteristics between the two grapes types. So what I'm showing here today, all the grapes are dots, right? So there's a good opportunity, you have an opportunity to taste these dots. So every year in the Geneva USDA repository, they have an open house. Normally it's in mid-September. This year, just look up. This year is in September 15, and it's a Saturday. They have an open house where you can actually go there, taste all the dots. I think you probably won't be able to taste the Muscardina grape because they're southern US. They cannot grow, survive in the winter here, but most others, I think you should be able to taste there. And it's a really fun event because you will be surprised to see how much flavor you probably never experience from the grapes you get from the grocery store. So next, I'm going to tell you about a story about grape domestication, mostly about sex locus domestication story. So this work we did a couple of years ago. It's an old story, but it's a very interesting story, and it's relevant to the breeding we are doing now. The current model for the sex determination in the grapes suggests that grape ancestor of the vitis, the common ancestor of vitis, is monoecious. Basically, male and a female organ on the same flower, and probably they're self-incompatible at that time. So basically, but it's monoecious. Basically, you don't have male and female separate, male and female plant. So that's a common vitis ancestor. But when the vitis genus, as a genus, when it emerged, it becomes dioecious. Basically, you have different separate male and female flowers. During domestication, which happened probably around 10,000 years ago, domestication select for the become monoecious again. So today, in the grape genus, in the Vitis genus, all the wild grapes are dioecious, and all the domesticated grapes are monoecious and can be self-fertilized. In 2020, a group in UC Davis, which Daryl Cantu's group, published a paper on Nature Communication, and basically they examined the male and the female locus of the genomes. And they identify a model to explain the grape sex locus development. So what they show is, so in the grape sex locus is a very small region, chromosome 2, which only has 150 kb region. So in this 152 kb, in 150 kb region, there are two genes. One is called YAB3, and the other is called IMP1. So their two genes are very close to each other. And so YAB3 is based on the homology, its gene expression is probably involved in the couple development, while IMP1 is regulated the pollen development. So one is for female organ and the male organ. So the ancestor for the grapes probably have a functional copy of both YAB3 and IMP1. So that's why they have the perfect flower, have both and perfect male and female organs. Then two separate mutation events happen in the YAB gene and the IMP1 gene, give rise to the separate male and female plants. And then we give us the Vitis, lead to the dioecious in the Vitis genus. Then during domestication, the recombination happened between these two genes. So they switch, the recombination crossover between two genes. In the wild grapes, we never have the two functional genes on the same haplotype. But when this recombination occurred, you basically restore the original state. You have two functional genes on the same haplotype. When that happened, we basically restore back to the monoecious status. So you get hermaphrodite flowers. So basically, domestication is simply during the domestication, there's a single recombination event give rise to this hermaphrodite flowers. And so basically, this recombination event is very rare, and then selected by the human ancestor identify this recombination event. They probably notice some very density cluster, and they start to preserve it and become of today's domesticated plant grapes we're used to today. So in parallel to what Daryl's work and Jason Londo in Cornell, and together with Chen Zhou in my group, they basically also they are looking at the sex locus. So what they did is they basically download all the public available data from the NCBI. They analyze the data. So in this plot, what you see in this plot, it basically is so Chen basically map all the reads to the both the male and female haplotype of the grapes. So if you look at the wild grapes, you can only see either female over homozygous F over F, female over female haplotypes, and which give rise to a female plant. Or you could see this purple colored is male over female, because male allele is dominant over female. So you get it, which gave you the male flowers. So basically, in the wild grapes, you only see female or see males. In terms of genotype, is F over F or M over F. But when Chen looked at the domestic grapes, so in domestic grapes, 100% of them are hermaphrodites. So what you can see here, there's a switch here from red to purple, which is where the recombination occurred. And what's interesting, what Chen was seeing is there are two distinct group alleles in the hermaphrodites. So in one case, in one instance, we call H1, which is same as I think Darius group identified. The recombination occurred at this location. And the H2, in H2, recombination occurred in this location here. So there are two independent events that lead to the domestic grape we have today. And these two alleles are basically both widely used in the grapes we have today, in both wine grape and table grapes we have today. So quite famous wine grapes are either H1 of H1 or H1 of H2, as you show here. So Chen also be able to, by looking, compare a sequence of this haplotype sequence, align sequence of this H2 allele, she was able to show most likely this recombination in the H2 allele occurred probably in the Caucasus region. So which is corresponding to an archaeological evidence that grapes originally was domesticated in the Caucasus region. So at that time, Chen was not able, because with limited availability of sequencing data, Chen was not able to identify where this H1 allele, where the other recombination event happened. So last year, there was a high-profile paper published on Science by a consortium of, very big consortium of European and Chinese groups. Basically, they show the second recombination event happened in the Iberian Peninsula in today's Spain. So basically, there are two recombination events, both selected by human, and they both exist today in the domestic grape we are using today. So one thing I would like to say is domestication is not just single event of this recombination. It's a very long process, and there are multiple genes involved in the flower development, not just this YABI and IMP1 genes. And there are other genes too. And I just talked to some grape breeders recently, and they said, if you simply just introduce this H1 allele into this white grapes, you not necessarily get the, like this good grapes we're used to, and they tend to be the cluster tend to be a little bit the cluster tend to be denser because of the self-fertilization, but berry also tend to be smaller. You don't get nice and tasty grapes. You simply just use this H-sex locus. Also, there's a long history in the flavor development, and this development is still evolving because the human's preference for the taste keep changing during history, and wines people like in the Roman time are very different to the wine we are used to today, from the wine we like today. Also, there's a seedlessness, which is a very newly added trait, and which if you go to the grocery store today, you probably, most grape we are getting seedless, which is the very latest addition to the domestication trait. So that's grape domestication. So there is it's getting increasing necessity to create this, to build this interspecific hybrids. So Vitis vinifera is originated from very small chimeid zone, which is in Europe. And today we have grapes pretty much we have domestic grapes that have been growing almost all over the world. So the driving force to create this interspecific hybrids, and one very driving force is the disease resistance, because the European grape in general, they don't do well in North America because all the fungal disease, mostly the issue is the fungal disease. There's a very famous hybrid, which is a Concord. Lots of probably already tasted. So it's a hybrid between vinifera and Nebraska. And this Concord grape is so widespread in New York State. It's almost like wild grape here. And they serve historically play a very important role for the early settlers when they moved from the Europe to North America. And this is one of the major grapes. They do really well, especially in Northeast America. So before I talk more about this hybrid grape breeding, I want to just clarify some of the concept, which is gene. And I want to talk about from breeders, how do we bridge between the breeder's gene and the molecular biologist gene. So breeder definition of gene is more like the original definition of gene dated back to probably Greg Mendel and Thomas Morgan's time. Basically, a gene is a functional unit. I would not call it chromosome, it's more like a linkage group. So it's a function unit on the linkage group, and which very, very rarely or very difficult break down this functional unit. So it's basically the function unit on the linkage group. So I'm going to talk about this RPV3 gene locus, and which is a widely used by breeders for downy mildew resistance. It's located on the lower arm of chromosome 18, and it's widely used for the downy mildew resistance by breeders. And this work is by Dustin Wilkinson in my group. So the candidate gene of for RPV3 has been discovered in this 2019 paper from Italian group, Fourier and the Morgante's group. Basically, they threw the transient expression system on the leaf, and they show there's two genes, TNL1 and TNL2 genes, are responsible for the resistance in the RPV3-1 locus, RPV3 locus. And both are the nucleobind leucine-rich repeat genes, basically NLR genes. And we know the NR gene tend to be very complicated, but they form big gene clusters. So they show these two genes in this cluster are responsible for the resistance in the RPV3 locus. Because Vitagen is a project mostly focused on breeding, so we are interested in developing markers for the RPV3 gene locus. So what Dustin did is he basically collect all the genome assembly, high quality genome assembly we can access to, and also including some of them, actually many of them here is assembled by our own project. So what Dustin basically collect all the genomes and slice out the RPV gene locus, then align them together. And he did the gene annotation separately, individual by ourselves, because we cannot rely on the original genome project annotation. The issue is when you do the fine mapping and when you do the genetic analysis, we have very low tolerance of false negative. We have pretty good tolerance of false positive. If you have false positive annotated gene, that's fine, but we cannot have pretty much zero tolerance of false negative, which we cannot afford that. So Dustin basically is annotate each genome we could have access to, and then align them together. So you can see actually this locus is very complicated. There are lots of different allele forms. So here are more than 10 different allele forms existing in the assemblies we have access to. And he groups them by this allele designation 3-1, 3-2-3, and RPV27, and a couple of others, which we have no designation corresponding to existing breeders classification. Basically, this allele is breeders allele ID based on their work with SSR markers. So what you see here, there are a lot of gene duplication. So that's typical like an R gene cluster. You will see lots of gene duplication events and lots of pseudogenes and also transposon insertion, deletion. So lots of we call genomic structure variation in this locus. So for the breeder's point of view is how do we distinguish these different alleles? So when you do a cross between two parents, right? We get progenies. So when a breeder do a cross, you're actually dealing with 4 different chromosomes, right? Two maternal chromosomes, two maternal genomes, and 2 paternal genomes. So you're dealing with 4 different genomes. So basically, we have to be able to distinguish these 4 haplotypes when we do a just simple 2 biparental families. So breeders have been traditionally using these SSR markers, which just PCR amplifies the simple repeat regions. The good thing about SSR markers is you get lots of long allele series because SSR markers is scored as the length of PCR amplicons, right? So you get, so for example, we can say RPV3 is the, so in the RPV, in terms of RPV3, look at breed has been in a combination of 2 SSR markers. Then, so RPV3-3 here is represented is a no amplification from the UDV 305 or the 221 base pair in the UDV 737. So the good thing about this SSR markers, you can get a long allele series. So if you see this table, you can see the allele size like 271, 297, and 290, 287. So you can see lots of different alleles. So the advantage of this different allele series, long allele series, allow the breeders be able to distinguish whether the individual I have is RPV3-1 over RPV3-2, RPV3-1 over RPV3-3 because of this long allele series. But then if you use the SNP markers, if you want to use SNP markers, the problem is if you look at SNP markers, only look at 2 different. So basically in SNP marker, if you look at the position, for example, we show SNP marker here. Even though at every nuclear position, you could have 4 different nucleic acid, ACGT, right? But in reality, if you look at pretty much all SNP markers we have today, in particular SNP marker, you can only get 2 alleles. So basically, you can only get 2 states. So in the 2 states is not enough for us to be able to identify, track any of. So if in the 2 family population, we get 4 alleles. So 2 states is not able to allow breeders be able to track these 4 states correctly, how these 4 alleles segregate in the progenies. So that's what we care about, which progeny carry RPV3-1, which progeny has 3-2. So these 2 states is not enough, right? So what we have been doing in the Vitagen, from starting from Vitagen 2 and now to Vitagen 3, is we try to using, instead of SNP markers, 2 allelic SNP markers, we start to switch to the multi-allelic haplotype markers. So basically in haplotype marker, what I mean by haplotype marker is instead of 1 single nucleotide, we basically use a combination of quite many different SNP markers located in 1 single Illumina reads. So basically, so nowadays when people doing the genetic, normally just map the reads to the genome, you get SNPs. But during this process, you actually lost information because these SNPs are linked in the original reads. But if you separate into SNPs, you lost this linkage information. And without this information, you basically, you fall back these 2 allele states. But if you use haplo markers, which allow you to get a lot more allele states and give you much power to track the different haplotypes. So since starting from Vitagen 3, we just, so Vitagen has been through these 3 iterations. And in the previous iteration, we mostly focused on the genes. And starting from Vitagen 3, and we got basically, it was USDA, our industry partners, would like us to more focus on the basic translational work. Basically, we start in our grant proposal, we would like to move from the genes on shelf, basically for gene discovery, move on towards like they call sticks in the ground. Basically, we want to make, so the focus starts shifting from research to the actual breeding. So this sticks in the ground is this terminology was going by John Martini, one of the Finger Lakes grape grower, and everybody likes it. And we actually put into the proposal. We want to shift to the sticks. So what I'm going to show you is our so first like grape breeding directions. So for the grape breeding, it's because a specialty crop. We know like wine is kind of still considered luxury products, right? So it's very different from major crop breeding like you do with maize or rice, this kind of major crop breedings. So when we do the grape breeding, we do need to preserve the domestication traits like the perfect flower, seedlessness. And it's very important. Also, we need to preserve familiar taste. Okay? And in terms of taste, people probably like some surprise, but not too much surprise. So the key point, we need to preserve the familiar taste. We cannot change it too much, especially at this point in terms of genetics. We don't still have good handle of taste, grape taste. We can work on grape disease resistance. We can work on the flower development, but we don't have a good handle of doing genetics on taste. So at this point, the only thing we could do is try to preserve as much vinifera, basically try to preserve the tradition as much as possible. In terms of genomics, we want to try to preserve the vinifera genome as much as possible. But we would like to bring in the disease resistance from the wild grapes, from the North America grapes and the wild grapes or Asian wild grapes. Then we can bring the resistant gene and also help with the climate adaptation. For example, like Finger Lakes region Finger Lake winter is really too cold for the vinifera to survive the winter here. So in Finger Lake region, so probably only some vinifera can survive, probably very narrow strip along the Cayuga Lake, but a little bit further from the Cayuga Lake, the winters are a little too cold for the grape to survive the winter here. So it's critical to make, it's important to make, bring cold tolerance genes into the domestic grape. So enable them to survive the winter here. So at this step, at this point, most grapers, pretty much all, I think I would say pretty much all the grape breeding programs in the US are doing market-assisted selection of the large effect QTL, just major QTL, large effect QTL. Just try to bring just small number of genes which give a large effect into the domesticated Vitis vinifera. Try not to disturb too much of the vinifera genome. And we start talking about genomic selection, basically try to be able to work with the minor QTLs or small effect QTL. By this point, majority breeding programs still doing market-assisted selection. So what I show here is just the current Vitigen 3 pipeline. And what we are doing now is we're basically doing these 3 steps. The first is the QTL discovery, basically identify new QTLs, which give us better resistance or the climate adaptation to identify new genes. Then we design DNA markers can track these QTLs, and then we release the marker to the breeders. And hopefully, so breeder would like to this past markers, basically SNPs. So as I said previously, SNPs is very difficult to work with in breeding because only have 2 allelic states. But if we map the causal gene of a QTL trait, if we know which mutation give us this particular trait, then we can use this SNP marker for the marker-assisted selection. The advantage of SNP markers is because it's a lot cheaper to genotyping and more accessible to the breeders. That's what they like. So the main effort of Vitigen project has been focused on develop DNA markers. So we would like to have DNA markers just not working for a population. We'd like to have DNA markers that work for the genus, the whole Vitis genus. In the first stage, the marker development has been, the first set of markers in grape breeding has been developed as a SNP array, mostly focused on vinifera. So this set of SNP array, I think European breeder might still be using it. So it's only good for the vinifera grape breeding. And because they don't cover enough diversity to work with the whole genus. Starting from Vitigen 2, we developed this marker system called ArchMP-seq marker system, basically. And this marker system is developed for the whole Uveitis clade, Uveitis subgenus. And this year, we start working with Breeding Insight to develop dot tag markers that work with across the whole genus, including Muscardinian. So both ArchMP-seq and Technology, basically, they are both commercial proprietary technology. What they're doing is basically PCR reaction. PCR amplifies the target region on the genome. Basically, we select the genome, select regions on the genome we want to amplify using PCR primers, and then we basically be able to run PCR reactions to amplify these target regions, then sequence these PCR products. The only thing is when you do the PCR reaction in a lab, you probably only include one set of primer in PCR reaction, right? But when we want to do genotyping, we would like to be able to sequence thousands of loci simultaneously. So basically, if you think about if you mix several thousand pair of PCR primers in one single PCR reaction, the primer just start to interfere with each other. You won't get up anything useful. So what these two technology, basically, they are just using their own proprietary method or chemicals, allow us to amplify thousands of loci simultaneously. Then we sequence this loci. And also, when you do PCR reaction, they have to make sure they can amplify evenly across this region, because you don't want one region you get more PCR product than the others, then you spend more money and sequencing this some region than the others. So they need to be amplified more evenly. So basically, we are using commercial technology allow us to sequence multiple regions simultaneously. And then in order to design PCR primers, and we start to think that we need to make sure these PCR primers has been a core genome. Basically, core genome defined in the genomic regions present in all different species on the grapes. And what changed when Chen did align all the genome together, she found that only 10% of reference genome is conserved across the varieties. So we design markers. It's all designed from the core genomes. So design from the core genome, make sure you don't get missing data. As much as they always get amplified in doesn't matter which sample you're using. So now we are moving on towards the Biotech, working with the breeding insight. Basically, we need to extend the core genome to include the vinifera muscadina in the design. So we have shown that using core genome markers give us lots of advantage, including minimize the reference genome bias, because we compare with the published PCA plot using the whole genome markers. If you look at if you use the whole genome markers, you see like in the PC2, this green dots are vinifera. You see PC2 is all about vinifera variation. But in our case, if you look at PC1, PC2, I believe our PC3, PC4, vinifera are still very tight cluster. So that means we basically have very little minimum. So this variation is all caused by reference genome bias, because analysis was done through using the vinifera reference genome. That's why we capture the variation in the PC2. But with the core genome markers, we almost have no reference genome bias. So again, we call SNPs from we don't call SNPs. We use the whole read as haplotype markers. So in this plot, basically we show the advantage of using these haplotype markers. So with SNPs, you only get 2 allele states. With SSR, their published papers show SSR average have like 22 allele states. But with ArchMP, we are getting up to in average, we get like more than 400 allele states. So which give us much better to accuracy when we want to map a reads to the proper haplotypes. So in our current pipeline, we are using the ArchMP, which is very cheap to do also. It's like less than $20 per sample. So we are able to use the ArchMP markers for map the QTL. With ArchMP, so the marker number we have is about 2,000 markers. So if you think about grape genome, it's 500 meg base pair long. So in theory, we should get 250 KB resolution. But practically, we normally get around 1 to 2 meg resolution. For example, in this RPV33 work that Chen is working on, she was able to using RGM-seq markers to map to a resolution of 2 MAC basis region. But 2 MAC basis region still has lots of genes in between. It's very difficult for us to identify the causal gene from the 200. Then in next step, we basically using the whole genome sequencing approach. But we don't need to sequence every individual in the family because they're long. So whole genome sequence is a lot more expensive than the RGM-seq. So, but we don't need to sequence every individual in the family. All we did, we just sequence the individual, we identify recombination in that region, then we do whole genome sequencing. Then she was able to map to the, narrow down the region to the 400 KB region. Then she mapped the gene into this insertion, this RPV3 locus, its insertion, again, turned out to be the NR gene cluster. So by after that, then we'll be able to develop gene markers for the breeders to track this particular QTL. So basically, we have a very cost-effective pipeline right now for the QTL mapping. So since 2019, we have mapped more than 10 QTLs have been published and using this platform. And also Chance basically is working on convert because we can narrow down to the fine map to the narrow down to small regions. Or Chance working on develop cast markers. So we start to work on that last year, and starting from this year, we will have more large-scale verification with working with breeder to the verification. So after that, we should be able to release this cast marker to the as a commercial platform, allow all the breeders simply just genotype and do selection more cheaply. And so today I'm only going, I already told you about genotyping side of the story, because if you want to do the genetic mapping, the other side is the phenotyping, of course, but I'm not going to go into that detail, but just want to tell you that the reason we were able to efficiently map the QTL is also because this robotic phenotyping system developed by Lance Cato-Davison group, basically called Blackbird, can do very accurate and high super phenotype. So the major challenge and the future directions about this project. So one thing we have to realize when we do this work is our big enemy is for the breeding project is actually the chromosome. We cannot break down the chromosome easily in the grapes, especially in the when you do the hybrid grape breeding. And it seems like this is a problem pretty much all the breeding programs. We just cannot have enough recombinations. So in this plot, I just show lots of the physical to genetic map plot. You can see, probably cannot see the number here, but just want to tell you. So in average, we only get a half recombination crossover per chromosome. So in this plot, you cannot see the number, but we only get, normally we get a centimorgan number plateau at around 50. So we just don't have enough recombinations. And we also, when we work on sex locus, which I did in the plot, I did not show the surrounding region, but the thing is, recombination is very difficult to happen in this sexual locus. So we don't know what caused this. We don't know what's the molecular mechanism behind this, but there's 50 kb sex locus. It's locked for the recombination. When you move outside this region, as shown in the bulk segregant analysis, when you move outside this region, you can recombination level back to normal. It's locked. So there's something going on in either genetics or epigenetics, or we don't know yet. So it could be genetics, epigenetics, or fitting is a gamete size. So there's something going on. So what we really like to do is basically understand the mechanism behind this recombination barrier. So this will help us, if we can get more recombination, which is a big benefit. We can reduce the generation time, can accelerate the breeding cycles. So this is really important. And also, right now, up to now, we are mostly using MAS, using the large effect QTL breeding, but the pool of large effect QTL is getting smaller. We don't have unlimited supply of large effect QTLs, and we need to learn how to work with minor QTL. So lastly, I just want to just quickly, I will go through a small project, that Chen is working on now, but it's just we only have some initial result that looks very promising. So the idea is we have sequenced that many vines. So I'm sure here we have sequenced more than 66,000 vines. So the question is whether we can do something like what 23andMe is doing for a human, right? So you're probably familiar with 23andMe. You give your DNA and we'll tell you where your ancestor is, whether you have a distant cousin somewhere in Europe, in Germany or somewhere, or whether you have the some other DNA inside you. So what Chen has been doing is we basically started working on, in order to do that, we need to solve some technical issues, basically when it's phasing, error correction, imputation. So what she has been doing is to basically develop a method to do phasing, error correction, imputation. So at this point, so this slide just shows the verification result from her initial result and looks very good. Basically, if you look at this plot, we show the, so the phasing looks very clean in comparison with using the BioLeak, using Beagle-based phasing, which has lots of errors and also show lots of, is overrepresented by vinifera, imputed to vinifera haplotypes. So we think what happened could be is like you say I'm Asian DNA, I send my DNA to the 23andMe because 23andMe database is dominated by the European origins. So it will not work very well with Asian DNA. So that's probably the case here. But if you use the RGMC case with the same database, we get a much cleaner result. So we think it's working, and we probably try to move more in this direction. So if that works, so which is really nice, so we can breed just to very low pass sequencing, we can identify the recombination breaking points, and we can basically impute the whole genome. So in summary, what I told you about is we have vast of gene pool from 20 million genetic diversity can be used for improved the cultivars. And our major challenge, really, one of the biggest major challenges is mice recombination barrier. And also we need for the new breeding strategies. And so what I told you today is about we developed this genus-wide genetic marker system for vitagen breeding pipelines. And we basically use the core genome and we rely on the haplotype markers. So I'm going to stop here. And so I want to acknowledge people. Most, I said, so the work mostly is a collaboration with Lance Cato-Davidson. And most of the work I talked about today is mostly done by Chen Zhou and Dustin Wilkerson in my group. I think I will stop here. Sorry about. Yes.
Yeah, you mentioned a great talk. Thank you. And my quick question, perhaps you already clarified, I missed. So you are talking about genome sequences of, a lot of those cultivars generally grafted, right?
Right.
So you are talking about the silence genome sequence or just stock sequence? Silent 就是when you're grafting, right? When you do grafting.
Oh, yeah. No, yeah. So at this point, we're not talking about root. I didn't cover any of the root stock. I didn't cover any of the root stock breeding. It's all about sign.
Okay.
It's all about sign. So we are not, right.
Like disease resistance.
Right. So now we do have some, actually we do some have breeders doing rootstock breeding, but I think majority of the data we are using for this project is sign.
Oh, okay.
Sign breeding. So yeah, so there's some rootstock breeding, but not majority here.
Thank you.
Yeah, because I think the thing that with the rootstock, you can use the white grapes. It's a sign that we need using the vinifera, right? So we want basically bring the resistance back. Yeah.
So most grapes are vegetatively propagated.
Yeah.
I
So
wonder what it does to like the allelic makeup, because so breeders make crosses, right?
right.
But how effective is selection if you go to so they make crosses and they right away go to vegetative reproduction, right?
So vegetative reproduction is about propagation and growth. So the breeder, when doing the breeding process, is still mostly through crossing.
I think what it says are these three. at the
Yeah,
F1,
so-
or are they going to?
Yeah, yeah.
Oh, okay.
Sorry,
Okay. No, they
Just
screen at
for
F1. We screen at F1. So what we do is basically in F1 because grow a vine it's expensive. So we basically most of the breeder doing is basically we do the they get F1, then they basically get F1 population, then we do the genotyping at the seedling stage, then we do the prediction and they decide which one they want to grow, put into the field. Then for the QTL discovery no, there are no vegetative propagation involved. It's simply just
Yeah, so
yeah.
but for breeding programs for the
Yeah.
micro-assisted selection, it's not really multiple generations. Like
It's multiple generations, but it's more like, say, we cross two parents, then we get progenies, then they probably want to add some other gene from another parent, then they add another parent in the breeding program. So they basically keep adding more genetics into the component into your line. So yeah, so basically each generation is a small number of progenies, but they keep adding new material into their breeding program. So that makes things very difficult to do like genomic selection kind of approach. And also marker need to be, that's why we failed in the first when you SNP, basically when across 2 or 3 generations, your SNP lost prediction.
Do you have the resolution to see if their recombinations occur in the hemizygote regions, or are they only?
So in the hemizygous region, we cannot have recombination, right? We can only see the edge. We don't get, so hemizygous basically deletes, you lost one allele in the genome. So we have find very difficult, yeah, so there's some recombination inhibition around the hemizygous region, be difficult to combine. Yeah, so there's so that's why we'd be really interested in working with Wojtek to identify the mechanism of barrier of the recombination. So semizygous is semizygous probably simply due to structure variation inhibitor, but there's some other mechanisms we don't know yet. So we really like to be more systematic study of the recombination inhibitor, which I think it's not just grape breeder interesting. Have this issue like apple breeders, pretty much most of the horticulturist crop breeders have this issue. We don't have enough recombination. Simply, we just cannot. So that without recombination, also we cannot fix the genome because we talk about vegetative propagation, just leave lots of scars, lots of deleterious mutations, which cannot be easy to get rid of. So that's one big issue in breeding today. So sexual recombination is critical and needed. Okay.
Thank you.
Okay. Thanks. Thanks for the time.
This has been a production of Cornell University, on the web at cornell.edu.